How are you? Welcome to my YouTube channel D&D Pathshala. In this video, we will discuss regarding the chromosome and its structure. So let's get started with our video. So here in this video, we will be learning up to the general characteristics of the chromosome, and in the next video, we will learn from the detailed information of the chromatin, centromere and telomere, and to the human chromosome and the types based on the position of the centromere. So let's get started with our video. Now coming to the basic information of the chromosome, as we all know that there is a long stretch of DNA in a coiled type of manner. So it is coiled in such a type of way, means it can fit into the tiny nucleus. So this process that which leads to the chromatid structure. So this chromatid structure will be like a thread type of mesh. So this thread type of mesh will be present in the active type of metabolic cell, and during there the cell division will occur, and when the cell division occurs it will gain a structure. That structure is known as the chromosomes. Now coming to the history of this chromosome, means this was coined by the W. Waldeyer in 1888, and these terms, that is the chroma means colored, soma means body. So the derivation of this chromosome is chroma means colored and soma means body, that is the colored bodies are present, and these colored bodies are also defined as a highly organized type of order arrangement, means it is a type of sophisticated type of network arrangement, like it will be present in our body. So it is a sophisticated network type of management or network measurement in our body. Now coming to the definition of a chromosome, means it is an entire chain of DNA, and when coming to this entire chain of DNA along with a group of stabilizing type of proteins. So this is the entire chain of DNA which will be having a stabilizing proteins group. And coming here, these will be containing a number of many genes and hereditary type of units, and also regulatory type of elements, and also the nucleotide type of sequences. So these nucleotide sequences and these are the other elements like hereditary units or genes and regulatory elements. And this chromosome is also a very tight type of coil, means this is very tightly coiled around the DNA, and these chromosomes will be also containing the DNA bind proteins. So the DNA bound proteins or bind proteins will be help in the packaging of a DNA, like a packer and a mover. Our product will be delivered with a very great facilitated type of packaging. So here this chromosome will be also activating and the other type of proteins like the DNA binding proteins. So these DNA bound proteins will help in the packaging of the DNA, and it will control its functions. Now coming to here, for a general type of question, even if you want to untie, means if you have an idea to untie a chromosome and know how much length it is, so to open up this chromosome we have to see the chromatid type of thread will be present in this chromosome, means the chromatid type of thread like fiber, it is made into one type of chromosome. So this chromatid type of thread like fiber, if we observe in a very keen manner, means in a very proper type of manner, then there will be actually a collection of proteins. So these collection of proteins are actually known as histones. So these histones will be wrapping this with a string, means these histones are wrapped up by a string like structure, and that string like structure is the chain of DNA. So see here, from a small element to a bigger chromosome it is being evolving. Now coming here, it is extremely a millions of long of nucleotides, means a millions of long nucleotides are being present, and a hundreds of genes are present along its complete length. And if we discuss in a normal term, so the chromosome, means if we discuss in a general type of term, the chromosome will be looking as a long and a loose type of structures, like noodles type of structure it will be having this chromosome. So this is the basic information regarding the chromosome. Now we will move on to the other type of slides. Now coming to the structure of the chromosome. So when coming to the structure of this chromosome, actually we all know that the cell cycle is being consisting of the prophase, anaphase, metaphase, resting phase. So in this, Here mitotic type of metaphors will be consisting of this two type of symmetrical type of structures. So this chromosome that which actually consists two symmetrical like structures means, so they are known as the chromatids. Now coming to these chromatids, these are actually of made up of like each chromatid is made up of a single type of DNA molecule. Now coming to a single type of DNA molecule, these both type of chromatids that which are actually attached to the each other. So these chromatids that which are actually attached to each other, they are being consisting of the centromere. Now coming to the centromere, so we will discuss in the next video what is this chromatid and what are the centromeres and telomeres. For now, here the chromosome will be consisting of two symmetrical type of structures, that is the structures that are known as the chromatids. So chromatid will be containing a single type of DNA molecule and both chromatids will be attached with the help of a centromere. So these will get separated at the beginning of the anaphase, means at the cell cycle. So now coming here, these will be like a bead like type of accumulations. So these centromere bead like accumulations will be present at the chromatin material. So bead like type of accumulations are known as the chromomeres. So these chromomeres are actually visible sometimes during the interphase. So during this interphase, and these chromosomes are also present at the tightly folded type of DNA regions. So these will be becoming very prominent type of polythene type of chromosome. So polythene type of chromosome means it will be actually having like example of the giant type of chromosome and chromatin, that which is actually bearing with such type of beads which look like a necklace, or beads means it will be looking in the shape like necklace type of beads on the string. Now coming here, the chromatin. So chromatin means here it is a type of macromolecule or a genetic type of material we can say. So this is being consisting of the chemical composition, like it is consisting of DNA, RNA and the proteins. Now coming here, it will be helping in the formation of the chromosome in the eukaryotic type of organism type of nucleus. So these are the chromatin type of structures and the chromatin compositions. Now coming here, the centromere will be consisting of the specific type of DNA sequences. So these specific DNA sequences will be also consisting of special type of proteins, like these will be forming a disc type of shape. So these disc type of shapes are also known as the kinetochores, and when observed in this electron type of microscope, we will be seeing it in a form of like a plate or a disc type of shape, and its length will be in the diameter range of 0.20 to 0.25 nanometer range, and upon the primary type of centromere, these are present, that is the kinetochore is present. And the organisms that are having their chromosomes with one centromere, those are known as the monocentric chromosomes. If they are having two, then it is called as bicentric, or so these microtubules. And for some type of species, the centromeres will be fused. So along with these micro type of tubules that which are attached to the length and to the holosome. So this holocentric type of chromosome is holosome, I will be saying. So these are attached actually to the length of the chromosome. Now these are the points. That is the monocentric chromosomes means it will be having a centromere with one chromosome, means one chromosome will be having a one centromere. But in some species, if the centromeres are fused along with the microtubules, then those are said as holocentric chromosomes. So the holocentric type of chromosome examples are like for the Ascaris megalocephala chromosomes are holocentric type of chromosome. And coming to the telomere is nothing but the chromosome's end, means it will be preventing from sticking or with the other type of fusion with the other type of chromosomal segments. Now coming to the besides, like the primary type of constrictions or due to the primary type of centromeres, the chromosomes will be also possessing the secondary type of constriction, means so it will be showing its limit at any point of the chromosome, and they are actually constant in their position and also to their extent, and these constrictions are actually helping in a identifying type of particular chromosome in a set. So these chromosomes are having, and the centromeres are having their constriction at some point of time, and they are also constant in their position, and their constrictions means their limitations will also help in identifying particular type of chromosomes. Now these chromosomes will be also consisting of the nucleolar type of organizers. So these nucleolar type of organizers means these are having certain type of secondary constrictions, like they will be having genes that which will be coding for like for example 5, 8s, 18s, 28s ribosomes, RNAs. So they can induce the reaction by the formation of the nucleoli. Now by the chromosomes that which will be bearing round and elongated like knob like appendages, then those are known as the satellite types of chromosomes. So this is the structure of a chromosome. So these are the images. So here we can be seeing, so these are the two identical type of chromatids. One will be the exact copy of the other, that is of the DNA type of molecule. The next one, this is the P arm. So P arm means it will be having a short arm structure. Q arm will be having the long arm structure. So as I am discussing that this is the centromere. So centromere is nothing, that is the second part or the center part of the chromosome. So it is a constricted type of point, and if we untangle this chromosome, it will be becoming like a thread type of fiber. So these are the DNA type of molecule. So this string is the long string like DNA type of molecule. It will be having a compact structure of proteins that which are also known as histones. Now coming to here, as the in the metaphase, these are the dupe, means these both are sister chromatids, means having the same type of character. So these, as I have said in this picture, these are the P arms and the long arms are Q arms, and this is the centromere. So when this is fused, that is the diffuser type. So these are the sister type of chromatids. So this is the image. So these are the sister chromatids, and here what are the difference between the sister chromatids and daughter chromosomes? Means here, if it is consisting of this one chromatid and if it is duplicated, then these are the duplicated type of chromosomes, means if by the mitosis process and by the centromere. So if these are of same type of length and same type of size, then these are the sister type of chromatids, means with one and duplicated. And if it undergoes mitosis, then two type of chromosomes are evolved, that is one, these are known as the daughter chromosomes. Now coming here, what is the differences between this chromosome and chromatid means? So chromosome means it is a condensed, means it is a small form, means it is the most condensed form, means it is one of the most type of form that which is available of the DNA. Whereas when the chromatid means, chromatid is less condensed, means it is of a less important or less type of model than a chromosome. So here chromosome will be consisting of either single stranded or double stranded type of DNA molecule. So here in the chromatid, it will be containing two DNA strands. It will be joined by the help of the centromere. Chromosome will be like a thin ribbon like structure. Here the chromatid will be like a fibrous structure, that too long. Homologous chromosomes means of the same type of character chromosomes are not identical, whereas the chromatids, sister chromatids, are identical. So here it appears in the metaphase and the chromatid will be appearing in the interphase. And here the chromosome action means it will be involved in the distribution of genetic material. Here it will be involved in the metabolism of other activities of the cell. So these are the chromosomal type of materials like DNA or the RNA. Here it will be adopting the different type of structures, like it is circular while measured or while packaged inside the virus particle. So chromosomes will be actually varying in number, like the depending upon the structure of the organisms and even the number of chromosomes. The species type of number. So these chromosomes in each species will be also constant. So actually the chromosomes function means it will be acting as a physical type of structures, that is it is acted as a messengers of the heredity, means of the character inheritation. Now coming here, the size of this chromosome will be as a shortest, means the shortest human chromosome will be containing 4.6 into 107 BP of DNA, and it is actually equivalent to 14,000 microunits or micro m of extended type of DNA. And its most condensed stage, means it is the most condensed type of form, will be during mitosis. It is of 2 micrometer long. Now coming here, chromosome will be consisting of different types of parts in it, like they will be consisting of euchromatin, heterochromatin, chromatids, centromeres, telomeres. Now coming here, chromosome number. Chromosome number, as I have said in the previous pictures, there will be actually 2 copies of chromosome present in the somatic cells. And when coming to the number of the unique type of chromosomes, that is represented in the capital L, in such type of cell it is called as the haploid number. And the total number of chromosomes, that is 2n, it is known as the diploid number. So this ploid means the chromosome sets. So here the haploid set of chromosome is known as genome. For example, in prokaryotes, means prokaryotes will be having one type of cell. So eukaryotes will be having different types of cells. So when coming to the prokaryotes, chromosomes will be in circular type of shape. Eukaryotes will be having linear type of chromosomes. Means linear type of chromosomes means in the straight type of chromosomes. So eukaryotes also will be having these chromosomes not only in the nucleus but also they'll be having in the mitochondria, chloroplast. And coming to the number of the chromosome in each somatic cell, it will be same for all the members in the given species. Like for example, in the chromosome or the organism that which is having the lowest number of chromosomes and is the nematode. That nematode is Ascaris megalocephalus univalens. So this is having only 2 chromosomes in its somatic cells, means total 2n means it is equal to 2. So these are the number of the chromosomes that which are actually present in different organisms, like in maize 20, common fruit fly 8, earthworm 36, elephants 56, human 46, donkey 62, dog 78. So the most of the highest chromosomes are present in the goldfish. Goldfish is having 100 to 104 chromosomes, and in the tobacco 48, and also in the oat, that is the 42. So they are divided upon their dependent family like diploid, hexaploid, tetraloid. So these are the different type of organisms of the chromosome number. Now coming to the prokaryotic and eukaryotic type of chromosomes, as I have said. So the prokaryotic and eukaryotic chromosomes means here these are the differences. So there will be double stranded DNA in prokaryotes, whereas long stranded DNA in eukaryotes. So here single chromosome means prokaryotes, several chromosomes means eukaryotes. Here it will be occurring in nucleus and these will be occurring in nucleus. So there will be no homologous in prokaryotes. There will be at least a type of pairing type of homologous chromosomes also. So from the inside, and here in the prokaryotes it will be not having telomeres or centromeres. But whereas in the eukaryotic chromosomes there will be having telomeres and centromeres. Here and also when coming to the prokaryotic types, so these are actually organized into operons, and here these are the individual type of structures. So these are some of the differences, and these are the chromosomes and attachment sites, and these are the binding type of proteins. So these binding proteins is the cross section. So this is the DNA part. Eukaryotic chromosomes means this is the chromosomes and these are the nucleus. From that these is the strand. So these are the chromatins, histones, DNA. Now coming to the autosomes and sex chromosomes. As we all know, there will be 23 pairs of chromosomes in a human being. The 22 pairs of chromosomes will be every type of same, means these 22 pairs of chromosomes will be same in each and every human being. But the deciding pair of their gender, means the gender deciding pair of chromosomes is the last pair. So that is known as the autosome or allosome or the sex chromosomes. So here in the diploid type of cell there will be actually 2 types of chromosomes. That is the homologous type of kind, means it is not known as the sex chromosomes. Here, as I have said in the previous point, 23 pairs, so 44 chromosomes. So 22 are homologous, means 44 are homologous, 23, that is 23 into 2, 46. So these 46th, 45th and 46th chromosome will be the gender deciding pair. So if the homomorphic type of pair or either the heteromorphic pair, so here if the ovum and the sperm will be deciding to what type of chromosome it will be carrying. So if the female, means if the human female is having 22 pair of homologous chromosomes, that is known as the autosome, and the allosomes are, as I've said, 23rd pair. So if the homomorphic pair, means if the chromosome is carrying X and X, it is known as homomorphic pair, that is the 23rd pair. And a human male will be having their 23rd pair as a heteromorphic pair, means they will be having X and Y. So these are the sex chromosomes. So these are the female type of chromosomes, that is the X and X, whereas these are the male chromosomes, that is X and Y. Now coming here to the general characteristics of a chromosome. As I have said in the previous slides regarding the chromosome, this size of this chromosome is actually measured in the mitotic metaphase, that is in the cell cycle, the metaphase mitosis. So when the mitosis process occurs, as I have said, it will be of 0.25 micrometer in birds, to as long it will be up to 30 micrometers, and in humans it will be present in 5 micrometers. and the organisms that are having less number of chromosomes, they will be containing large type of size chromosomes. Means the less number will be having large size, the more number they will be having small size. So here even the shape of the chromosomes will be also changing the continuous phase of the cell division and also the cell growth. So during this G0, that is the resting phase, and also during the interphase, so as they will be like a thin type of coiled type of substances and elastic type of threadable substances. So these are like a stainable type of structures, and even this chromatin threads, they will be become thickening, and type of thickening process will occur and filamentous, during which phases anaphase and metaphase. So these are the general characteristics of a chromosome, and that's it for this video. And in the other video, means in the next video, we will be discussing regarding a detailed information regarding the chromatids and telomeres and also the telomere replication. So that's it for this video. If you like my video, like, share, comment, and please don't forget to subscribe to my channel. Please subscribe to my channel and please support me. I wish all your support and all your blessings are with me and on my YouTube channel. So please don't forget to subscribe my channel and please support me, and thank you.